right, we'll get started now for our final project summary. Today we welcome Nancy Ruiz from the Naval Facilities Engineering and Expeditionary Warfare Center in Port Hueneme, California. And she will be giving a presentation with her co-PIs on in situ wetland restoration demonstration. This was an ESPCP project number ER-200825. Before we get at this time, I'll turn it over to Nancy to get started. Thank you, Nancy.
Andrea, this is John Blyler. Nancy might be late. Nancy, are you on the call? I'll tell you what, why don't I be Nancy, and then she can join us.
Okay, John, thank you.
Of course. I know she had back-to-back calls going.
I've always said you'd make a great Nancy, John, so it's nice to fit into that role now.
I'm a father of 3 daughters. I'm used to it. Well, thank you, everyone, for joining. This is John Blyler. I'm with an organization called AECOM. We're co-investigators on this. Just before I get going here, can everybody see that I'm changing slides? I want to make sure I understand the software. Is the project team slide up and running for people?
Yep.
Terrific. Thank you. So Nancy, I know, had back-to-back calls and will be joining us shortly. She's been the principal investigator. Nancy actually transitioned from another individual, Amy Hawkins, who we were working with earlier on this project. Kevin, Dr. Gardner from the University of New Hampshire. Myself, Trudy from URDIC, and to a slightly lesser extent, Mark and Dave Tarcliffe are also engaged in this program. So, wetlands, as I think everyone on the call knows, often can serve as a sink for environmental contamination. There's millions and millions of dollars of environmental liabilities outstanding in wetlands. NOAA and others have done some work indicating that 10% of the nation's sediment is contaminated. It's likely that hydric soils within wetlands are in a similar position. And the study we're going to share with you today, I think, presents some opportunities for some significant sort of sustainable remedial alternatives when it comes to dealing with contaminated wetlands, as well as perhaps some more cost-effective remedies for dealing with wetlands as well. So this is an activated carbon study, but rather than working in subaqueous sediment, as so much of the work's been done, we worked in a marsh wetland system and asked some very simple questions relative to the use of in situ technologies and activated carbon and the sequestration or reduction in bioavailability of contaminants within the wetland. We, as I'll get to, had a number of different metrics for success following carbon application into the system. They included ecological and risk reduction methods, cost effectiveness, and also ultimately community and regulatory acceptance. I'm not going to spend a lot of time. These are the slides Nancy was going to present on the performance objectives. I think in retrospect, we might have even had a few too many. I've got 2 slides of performance objectives here, but they basically centered around ecological, toxicological, bioaccumulation type questions. So we were asking, once we've applied carbon to this wetland system, using a variety of different treatment technologies, pelletized as well as slurried carbon, what effects did we observe in terms of changes in pore water concentration of PCBs? What effects did we observe in terms of residual tissue burden levels of PCBs? We looked both relative to time 0 for multiple endpoints as well as to several different types of controls. So we had pore water, we had PCB bioavailability type endpoints using tissue analysis, and also a number of different ecological type endpoints looking at the macroinvertebrate community, looking at the plant community pre and post application. This next slide is sort of more of the same. It's more of the performance metrics that we looked at. Safety was a big deal here. The site we ended up working on is a UXO site. All the work we did required UXO clearance. It was not intentional. That was just the way it ended up via the site selection process. But in retrospect, these sorts of in situ remedial technologies may well lend themselves to UXO sites and other sites where there are sort of safety hazards associated with use of yellow iron and other traditional remedial technologies. So our program was fairly straightforward. We went out prior to application, time zero, collected quite a bit of data in the months prior to December 2010 into 2010, and then had 2 rounds of monitoring data. Ultimately, I'll give away the punchline. I think more monitoring is needed on this program. The data, the results are encouraging. I think we did see some very positive trends, but as you can see, it was a rather tight monitoring program. The program got delayed for several reasons, and we were running a year late into the program, largely because of environmental permitting needs. And so the monitoring program got truncated a little bit. But we had monitoring programs that occurred at all 3 monitoring events, time 0, time 1, time 2. They included chemical, bioaccumulation testing, ecological evaluations, physical evaluations, and also just sort of looking at the wetland itself to make sure that there were no sort of immediately apparent community impacts as well as more subtle impacts. What did I do here? Keep forgetting to use these gray arrows, sorry. Okay, so the site itself, we ended up working in a portion of the Aberdeen Proving Ground in Aberdeen, Maryland. This is a large facility, 72,000-acre Army base on the western shore of the Chesapeake Bay. It's a federal Superfund site. The area we worked is known as Canal Creek. Canal Creek is about 2 miles long or so. It has approximately 100 acres of wetlands associated with it. It drains, let's see if I can make a cursor magically appear here. It drains towards the bottom of this figure here into the Gunpowder River and eventually into Chesapeake Bay. Benthic and freshwater fish community are present at the site. For decades, municipal and industrial wastewater was discharged into this area, and as part of the CIRCLE program, the risk analysis demonstrated potential risks to the food chain, the benthic receptors as well as cruciferous birds, mammals, wetland ecology. I think the food chains they used looked at raccoons, herons, kingfishers, and so forth. This was all work done by others. PCBs and DDT were our focus. There's also a portion of this river and the associated wetlands that are mercury contaminated, as well as some other metals. So we felt it was an ideal site for a variety of reasons, one of which is that the footprint of these environmental constituents doesn't overlap completely. So there are portions of the river that are heavy in PCBs and relatively clean compared to some of the other constituents, and then there are other portions of the stream system that contain mercury, contain DDT, but don't contain PCBs. Important to note that this was a collaborative opportunity in the funding cycle. 2 projects were funded. Charlie Menzie and Upal Ghosh had a project downstream from us. We worked in the upper portion of Canal Creek. They worked in the lower portion of Canal Creek, but somewhat similar or complementary project objectives. I'm not sure if this group has heard from ER-200825 or not. Okay, the PCBs in this portion of the system were well characterized, well quantified. This colorful map and the various dots simply represent different levels of PCBs within the system. Red is up to 100 ppm, many blue dots, blue triangles, different sampling events, squares representing 1 to 10 ppm approximately PCBs, and then some lower levels. To give you a sense of scale, I think we're looking at about 5 acres here in terms of the shading on the marsh system. One of the objectives that we started with, we eventually moved away from it a little bit because it didn't seem like there were any readily apparent results, was looking at this blue cross-hatching, this red cross-hatching. In a sense, 2 different systems. The creek is off here to the west. To the left of the figure, there's a higher value wetland system, an emergent marsh system, arrowhead, RM, a wide variety of emergent plants, fair diversity. I think we had 49 species of vegetation counted in there. And then we also have almost a monoculture of Phragmites in this red area. So we tried to compare and contrast. We ended up collapsing the data into one data set rather than 2, but that was part of our objective. What else? PCBs originally were quantified as hour chords. on this site, primarily 1016, 1248, 1254. We moved away from that into a congener profile, so the PCB data that I'll be presenting here and after are total PCBs based on congeners. There were, we looked initially at all 209 congeners. There were about 29 of them, as I recall, that accounted for close to 99% of the total PCBs. So the 29 congeners was where we focused our efforts going forward. See if I can make my green arrow go away. Initially, we looked under ideal laboratory conditions at both DDT and PCBs. We collected treatability study data, brought soils back into the laboratory to do some bench scale testing, looking at bioavailability parameters pre and post treatment with activated carbon, looking at pore water, looking at bioaccumulation studies. The effectiveness was in essence determined based on reduction in hydric soil and soil pore water concentrations, again with the PCBs where we ended up focusing. The 29 congeners is what we focused on. We looked, I have a data slide, is the next slide, but we looked at a variety of different amendments initially. We settled on activated carbon, but we also looked at organoclays and zero-valent irons as part of the initial pilot study work. And the study indicated that 3% carbon was a good way to go. This is incidentally a fairly common number. A lot of the subaqueous sediment activated carbon amendment pilot study work and even demonstration scale implementation work tends to settle around 3 to 5% activated carbon in the sediment. What we're looking at here is the percent reduction in pore water of PCBs. AC3 is activated carbon at 3%, AC6 is activated carbon at 6%, organoclays and zero-valent iron as we proceed to the right of the chart. This is a few of the congeners. These were 5 of the predominant congeners, but as you can see with 3% and with 6% activated carbon, we didn't see much difference, so we felt we could pretty well optimize on the 3%. Organoclay was slightly effective. We did see some pore water reductions in zero-valent iron. In fact, the opposite we saw what likely represented liberation of specific congeners via the reductive dechlorination. So they were probably congeners that were stuck to soil particles and were actually liberated into the interstitial pore water.
And John, just to point, those zero-valent irons were really included for the thinking about the DDT as a possible remediation measure for DDT, but obviously not very effective for PCBs.
Very good. Okay, part of what we tried to do here is, we had no ownership of any one technology or any one delivery system, so we were tasked with evaluating several different amendments in a comparative fashion. We included 2 different pelletized amendments, aqua block, which is in essence a small rock or a stone coated with bentonite, and the bentonite is coated with carbon, so we use that as a carbon delivery system. So it's an aggregate-based system. We again collaborated with Charlie and Upal, the 835 group, on the sedimite program. So we had both aqua block and sediment data to compare and contrast. Those were both dry broadcasted, and then we also worked with a slurry, powder-activated carbon slurry. One of our goals here was to try to use readily available landscaping equipment, nothing too fancy. What we ended up doing was renting a hydroseeder and using a large sort of industrial hydroseeder with a mixing tank to mix the carbon slurry. And we actually played around with a bunch of different technologies and settled on a bark blower, another piece of landscaping equipment for the aqua block delivery system. And then the sediment was also dry broadcast with a backpack unit. But again, our goal was to try to use things that were readily available from Taylor Rental, those sorts of places. They weren't entirely pleased when we returned their equipment covered with black carbon, but we didn't destroy anything. It was just a little darker than when we got it. A lot of the data that we collected was evaluated by looking at control plots, unremediated plots, as well as control plots that we covered with a sand mixture. It was actually a sand-soil mixture, a manufactured mixture we mixed on site in an attempt to look at the native soil permeability and the native soil organic carbon. The reason we used that second sand control was actually in response to some very good comments from the ESTCP Review Board. And the question was, are we looking at just kind of a thin veneer of soil covering the wetland, and is that what's driving reductions in bioavailability, or is it actually the more expensive and more difficult to deliver or get activated carbon amendments? So we had several different types of controls that we used. This gives, again, a sense of the different plots that we had throughout the area, looking at time series data pre-application and post-application, as well as looking at different plots. That you can see we had, a number of different plots for sand, for aquablock, for slurries, for sediment. We tried to distribute them throughout the system. The only things we generally did without distribution was the slurry spray tended to go in the higher wetland. We were concerned about it washing away in the deeper water emergent marsh towards the left of this frame. It's about a 2-foot tidal flux at this site, so there is some, it's freshwater tidal marsh, so there is some tidal flux, and we were concerned that the slurry might move around a little too much, and so we kept that in the drier portions of this wetland system. This gives a sense of the site layout. This is looking towards some of the across what we call the high-value wetland, towards the Mighty's Marsh in the background. You can get a sense of scale. There are several workers there. We worked with plywood pathways. We did end up, because even though this was a surplus site, it was a non-circular activity, or so it was deemed by the regulatory authorities. So we did have to apply for an Army Corps permit, which, on the one hand we lost some time. On the other hand, I thought it was very valuable, because the Corps Baltimore District determined this was not a discharge of fill material into a water of the U.S. and we could proceed. And I think that's very good information for future studies of this kind. But it gives you a sense. We bounded our areas. Each of those squares I showed in the previous slide is about 8 meters by 8 meters. There are 24 of them. We bounded them with these Squire Fatine logs to try to eliminate carbon movement between plots. In retrospect, we probably could have gone a little bit higher. There were some lunar tide events that did overtop the Squire Fatines, and I do think carbon moved around a little bit in the system, which did make interpreting our data just a little bit harder. The channel depth, as you're going towards the actual center of the channel at high tide, can be up to 6-7 feet, and the tidal flux is 2 feet. So there's a fair amount of water moving around this system. These kinds of systems tend to be high in native organic carbon as well. It's probably also worth pointing out at times this is sort of a soupy organic matrix, up to 15 to 17% total organic carbon. The materials were delivered, again, based on the treatability study with the goal of achieving a 3% activated carbon in the field. This simply shows the dry broadcasting. This is roughly a 200-foot long hose that we ran from the arc blower, 4-inch hose, and it worked very effectively. We were able to deliver carbon well out into the wetland via this hose system. There were times that, if you could envision sort of a little bit of an elbow in the hose as you crossed hummocks, and the uneven topography of this marsh system, where the carbon would back up, and it took a little bit of coaxing to get it out of the system. We did do some pilot studies up front, just in a parking lot, to make sure delivery of aquablock through this 200-foot hose didn't result in the loss of any carbon. We didn't want to put in an aggregate coated with carbon at one end and deliver stones at the other end. And we were very comfortable that the aquablock survived the trip through the hose. And then this is the wand on a carbon slurry delivery. This is 30 to 50% activated carbon by weight. So sort of think wallpaper paste almost. It was a very thick slurry that we delivered to the marsh in this system. 300 linear feet of hose from the hydro seeder back to the upland. So we were able to deliver across a long geography, although it got cold and there definitely were some issues with winter delivery. So it's a lesson learned type thing. the monitoring, I think I've mentioned already that we looked at these multiple time events: time 0, time 1, time 2, UXO clearance at all sites. We also did some passive sampling, looking at PCBs and pore water changes with depth using passive samplers post amendment. Despite all of this monitoring, and again giving away the punchline a little bit, the carbon, as I think hopefully people will agree, did look like it worked. It may well be effective in these settings, but boy, there was a lot of heterogeneity in the PCB data set. We had 2 ppb to 200 ppm, so orders of magnitude differences in PCB levels, and small sample size. The mixing of the carbon in this kind of system is probably different than the mixing in a subaqueous sediment system, and as I've mentioned, we may even have seen some migration of carbon. So there were some confounding factors that you need to keep in mind as you look at some of the subsequent slides. Gavin, it might make sense to pass the baton to you for the next series.
Sure. Yeah. So this is Kevin Gardner. We had performance assessment were one based on just PCBs, just looking at PCB bioavailability, and other measures of ecological and plant health, as well as things like constructability and cost and so forth. On the next slide here, we detail a little bit more. What I'll be talking about at the beginning here is the performance assessment relative to PCB availability. And so we measured pore water by collecting soil samples and bringing them back to the lab and using palm extractions to measure pore water PCB concentrations. And we found, because of the large heterogeneity that John was talking about, and as we'll see in the results here in a moment, the results weren't very conclusive when we just looked at pore water, looking for those pore water reductions associated with having the highly sorptive activated carbon present in the system. So the other way that we did our performance assessment was to look at the partitioning between the solid phase and the liquid phase. So in essence, by adding activated carbon, what we're trying to do is to change that partitioning behavior so it partitions more strongly to the solid phase. So this is the way that we tried to deal with the tremendous spatial heterogeneity in terms of total PCB concentrations. So in this, at the bottom of this slide here, it shows how we evaluated those. They may be a little bit unconventional, but basically this partition coefficient is a concentration in soil divided by that in pore water. So with treatment, with activated carbon addition, that partition coefficient should go up, so it's partitioning to the solid phase. And the second one is the most unconventional one, but this is basically the same thing, partitioning to the solid phase. So in either case, with treatment, we should see a higher partition value, a higher K value. So this first slide here shows the pore water PCB concentrations. So here's concentration, and then we have the different treatments on the x-axis here, and then 3 different sampling events: time 0, 6 months after application, and 10 months after application. And I should point out, some of the things that John referred to caused some significant delays in the project, and so as a result of that, we have a technology for which we expect there to be treatment over a pretty long time horizon. That is, we were thinking that the mixing processes in these kind of soils would be fairly long term, and we're evaluating it over a really short term. And so that was a non-ideal kind of outcome on this project. But anyway, if you look at the AC slurry addition here, you see maybe some modest decrease, factor of 2 or less, in pore water concentration. The aquablock going from time 0 here to time 6 months, 10 months, perhaps some reduction about an order of magnitude or less. Sedimite, we didn't see any change in the pore water concentrations, and similarly, sand control in the control plots, we saw, pretty consistent concentrations of pore water, PCBs. And so this is what led us to scratching our heads a little bit and thinking about what role the heterogeneity of the soil in the PCB concentrations played in these results. And we have later there's a slide that shows which of these differences are statistically significant, and so we'll revisit that in a moment. So the next slide here shows tissue concentration in lumbriculus. So again, from time 0 to 6 months to 12 months, 10 months, we see some reduction in tissue concentration for the activated carbon slurry addition, the aqua block. We, because of that collaboration with Charlie Newpal, we didn't have tissue concentrations for time 0 for the sedimite, so we weren't able to evaluate reduction in tissue concentrations from this plot. And then sand control and control, we see no significant change in the tissue concentrations. So we kind of have a little bit of a mixed message here. We have maybe some reductions in pore water. We have some reductions in tissue concentrations. And so this plot shows partitioning behavior. So this is measuring PCB concentrations in pore water, measuring PCB concentrations in corresponding soil material, and this is the partitioning coefficient here. So what we see in activated carbon slurry and aqua block and sedimite is we see that there is an increase in the partition coefficient, it looks like. So this is the kind of behavior that you would expect. We're putting a highly sorptive phase into the soil. We would expect greater partitioning of PCBs to the sediment matrix, which now contains this activated carbon.
Yeah, but I might just add on the right here, you also see this trend where the control appears to be showing an increase in the partition coefficient, and that may well be associated with what I mentioned earlier, that the carbon did move around in the system a little more than we hoped it would.
Yeah, we did find concentrations of activated carbon in the control plots, and we can look at our layout and maybe reevaluate if that was the best approach to do that or not. But for sure, there was some dispersion of that activated carbon, and there were some big storm events that occurred in between these periods of time. I forget off the top of my head what it was, but I think there were 2 major rain events. One of them was a hurricane. Do you recall, John?
Yeah, there was a 100-year storm event that occurred.
And so then here's the partitioning again between tissue and sediment. So again, we would expect higher partitioning coefficient when we add activated carbon, and so that's what we see in the slurry, the aqua block, in the control. Maybe the sand control displays the same type of behavior at the 10 months, but a good amount of heterogeneity and lack of a really clear signal of treatment efficacy here.
John, Kevin, can I ask a question?
Yes.
This is Jason. You guys did this application during the winter, correct?
Yes.
And you cleared you cleared a lot of the vegetation during the winter?
Yep.
And so these storm events, did they happen before the growing season or after the growing season, so that your hydrodynamics, because of the vegetative growth, might have been different than they normally would have been? Do you understand what I mean?
Yeah, no, I'd have to go back.
That's why you might have had higher transport of your activated carbon out of your test plots versus a fully, grown. You had a growing season, and probably that occurred between point A and point B, and then you had probably less transport activated carbon after you had a growing season in the wetlands.
Yeah.
That may well be the case. We didn't, clear vegetation willy-nilly. We ended up doing everything we could do to avoid clearing of vegetation, in part because that's what our core permit required. But so we didn't put any permanent roads in. We put the plywood walkways that I showed earlier, and the vegetation was cleared only in the Phragmites plots, and no grubbing or anything like that. It was cleared to a foot, 18 inches after the growing season, just for ease of application. It was hard to get in there. The Phragmites monoculture was just so dense. But we knocked it down from 6 or 8 feet to 18 inches, and it's got a 2-foot tidal flux anyway, Jason. So you may well be right. And we did anecdotally. I have some photos of it here and there, but we anecdotally saw a thin, kind of a black carbon film on the fronds of sensitive ferns outside of a few of the plots. That led us to believe that was going on as well.
Yeah. All right. I just wanted to ask because.
Oh, good point.
All right.
We just to follow on, we have some evidence of carbon movement, and we have some evidence of really low activated carbon, in particular on the sedimite plot. And those are clustered in a different location where they could be subjected to higher shear stresses during 100-year storm type of events compared to some of the ones that are further upland. So we also have, we'd like to get back out there and do additional sampling at a more appropriate time horizon, like now, and we collected some cores, some sediment cores, at the 6-month sample application time, and we have those, and we've analyzed a bunch of those, and for sure, carbon distribution is a really important component that's going to help us explain these results. And in addition to that, the PCB concentrations vary significantly over depth. So they vary spatially, and they vary significantly over depth. And so it's really important in evaluating this to understand the depth profile of PCBs and of carbon, and of the possible migration of carbon off-site, so that we can nail down, how much of the results that we're seeing are influenced by, this potentially influenced by a big, large storm event like that in this particular site. So those are things that we'd still like to be able to do. So what this chart shows is where there are statistically significant treatment effects. And so we saw in those previous slides that there are some noted differences. In this slide, we see which of those differences are statistically significant. So the first column here is whether the post-treatment is different than the pre-treatment. The second column here is whether it's different than the control plot and changes in the control plot. The third column here is whether the partitioning is different than the pre-treatment, and the last one is whether the partitioning is different than the control. So these first two are whether pore water is different, and then whether partitioning is different. And then down here it's whether the tissue concentrations, the absolute value of the tissue concentrations, are significantly different. And over here is whether the partitioning of between tissue and soil are significantly different. So we see that the heterogeneity was so large that in a lot of cases, even if we think we might see some treatment effect, statistically it was not significant because of that heterogeneity. So John, I'll turn this over to you.
Thank you. And I see that Nancy has joined us.
Yep, finally. Ready Talk let me in.
Hi, Nancy.
Hi.
We'll just march along, but please dive in at any time.
Okay, thanks, John.
I won't spend much time on the community monitoring. The macroinvertebrate community surveys were not very helpful, in part because there just really wasn't much of an indigenous community at the site. Again, this is not a freshwater pond or river system where you're going to be looking for EPT and chironomids. So we made a good faith effort, didn't find much. That in and of itself is interesting, though, because as Charlie and others have pointed out, though, the macrovertebrate community in pure subaqueous sediment sites often serve as the engineers delivering the carbon to the subsurface. In this case, I think it's root elongation and plant growth as much as a limited worm community and annelid community. But we have a number of photographs of carbon sort of working its way down the root elongation pathways up to 6 inches below the surface and just moving right along with the roots as they grow through the season. They're really interesting photos to look at. Wetland vegetation, we looked at a number of different ecological community diversity, species richness metrics, didn't see any pre- and post-treatment changes. The only real significant effect we saw is as the growing season went on, all plots had more plants. So no great surprise. It's a little harder to identify some of the herbaceous material in the December time frame and in the end of the growing season time frame. And no gross effects in terms of early senescence, yellowing, stunting, withering, anything along those lines. We did, in response to the group, this wasn't part of our initial study, conduct a nutrient study. The concern was, we're adding a sticky agent, we're adding carbon to the system, is it possibly binding nutrients and inhibiting plant growth? So we collected intact cores from the different plot types, sent them off to the lab, ran a long-term laboratory study with the wetland plant Echinocloa, picked up a number of different toxicological endpoints by running that study, even though that wasn't the goal. We looked at shoot length, weight, mortality, did not see any statistically significant effects across the board from controls. We also looked at a number of different trace nutrients to see if they changed over the duration of the program. And the way we did this was very simple. We looked at an uptake factor. We looked at concentrations in plants versus concentrations in the hydric soil of these trace nutrients, and we compared them to the site control as well as the lab control. But these data are the site control data. An upward arrow indicates some enhanced nutrient uptake. The very few downward arrows that we see actually show the opposite, show a detrimental effect. So this was, interesting. It's not really what we expected, but if anything, there was a little bit of enhanced plant growth, enhanced nutrient uptake here. Looking in the literature, we're not alone in seeing this. There are some studies that have shown this before. So, different carbon forms certainly can absorb nutrients, but also they may stimulate microbial activity. So we may be seeing nutrient cycling resulting in higher availability of nutrients in a form that plants can use. There's also a body of literature out there that indicates that activated carbon in these kinds of systems can bind allelochemicals, toxic chemicals produced by invasive species, which can actually benefit the growth of plants. So the general consensus is the exact mechanism is not really understood, but other folks have seen these sorts of trends as well. Just in terms of the overall findings, the data, as I've indicated, I thought were encouraging. They suggest that activated carbon indeed is sequestering PCBs in the wetlands. There definitely is a temporal component that it seems to be taking a little bit longer in this system than some of the subaqueous sediment systems. To Kevin's point, additional monitoring over time I think would be very illustrative here. We've mentioned some of the confounding issues that we've run into at this site, but again, I think the overall findings were encouraging. We're not statistically significant, at least we were seeing trends in the right direction. In terms of the cost assessment, we did a fairly exhaustive analysis in our final report, and what we ended up doing was looking at sort of the costs of traditional remediation of these types of systems, and so often that's dependent upon the contaminant disposal option. I mean, if this was a TSCA level of PCB site, the costs go up and so forth. But typically $1 million to $2 million is a number that's carried in a feasibility study for these kinds of systems. We're looking at somewhere between $170,000 and $360,000, assuming a 1 acre project. There are definitely some economies of scales with bigger projects. What seems to be really driving so much of the cost assessment is mold demold. If you need to, some of the analyses we ran, if you had to put in swamp mats and really, build temporary roads to access the wetland, that costs money. And interestingly, monitoring was the other one. The sensitivity analysis, long-term monitoring, especially given that this technology isn't, it's kind of new, it's evolving, maybe not a high degree of regulatory acceptance, so there might be a requirement for elevated levels of monitoring and multiple plots, PCB congener analysis. So much of the cost is actually driven not by the amendments themselves, but by the long-term monitoring and the different sort of mold, demold assessments. This is a typical example of the kind of framework we presented in our report, where we tried to break the costs per treatment area down. And there are some economies of scale, whereas larger sites, the unit costs tend to go down. In terms of scale up, the technology is readily scalable. There are somewhere around 25 large-scale studies of this nature underway in the U.S., in Norway, and elsewhere. Some of them are in wetland-type systems. Some of them are in more pure subaqueous. The full-scale evaluation may require a little more pilot scale than we did, more post-treatment monitoring. Again, I think this is largely stakeholder acceptance-driven for now. That may well change in time. The slurry released carbon pretty quickly in terms of scale up. We think the pelletized material less so. Roots as a primary bioturbation agent also probably will take more time in terms of scale up and equipment considerations. I think some of the commercial vendors, Aquablock for instance, have really looked hard at using telebelts, using different dry broadcast systems, even using helicopters on certain sites. So I think there's a wide variety of scalable delivery systems one could contemplate on these types of systems. In terms of lessons learned, the material was readily deployed with the landscape equipment. I thought that was very positive. The movement of activated carbon was potentially a concern, and the dead stands of Phragmites were a challenge as well. I think the spatial and temporal variability, we thought we had a well-characterized site, and in retrospect, we probably could have done more. The substrates in the sort of the lower high-value wetland are very different than in the Phragmites marsh. And I think understanding some baseline conditions a little bit more than we did might be very helpful. There was some movement of the activated carbon, as we've mentioned. And the biological impacts I thought were quite manageable. Again, really no gross ecological effects were observed, and long-term monitoring is critical. I did mention the cold weather concerns. The slurry relied on an aqueous matrix. When it got below freezing, that was challenging for us. Available tools and training. One of the tasks that we undertook was development of a framework guidance manual. So this is a manual that's designed for end users of this technology. It's, Nancy, you can dive in here, but it's going to be published shortly, imminently. It's been completed. It's been approved by ESTCP. We're just waiting for some procedural.
There have been issues on the Navy's end to develop some acceptable standard disclaimer language associated with these reports. Because these are public documents and they're going to be on the public website, both on ESTCP and the public side of the Navy portal, there's been some intense scrutiny of these types of reports, and so there's been a lot of effort to get, as I said, standard disclaimer language, and we're looking extra hard at appropriate attribution of any images that are in there, just to avoid lawsuits against the Navy in the future. So my apologies for it taking so long. It looks like we're coming to a good conclusion very, very soon, but now we're working on the appropriate attribution of photographs and images.
And it's not a cookbook. It's a framework. But what it's designed to do is guide end users from everything through from sort of the site selection, amendment application, through pilot studying, treatment, reporting, permitting, right through to the monitoring, and provides, when it was written, I think the most current references and tools available. Ideally, it's the sort of thing that may well be a living document, but I think it's a good start, and hopefully will be a valuable addition. We've also, and maybe Nancy, you can take over here, but we've presented a number of different platform presentations, posters.
Yeah, in the wider technical community, there have been posters and platform presentations, and for the DoD community, there's been some specific addressing of our remediation technology, our ART work group. John and I believe wasn't it Charlie that you and he put together basically a combined presentation of the two projects to really give an indication of the breadth of the technology that was being applied in these environments and the success that both projects achieved.
And, it's probably worth mentioning here as well that there's been quite a bit of industrial commercial interest of late in this technology. I've been in front of two major corporate type entities in the last quarter, sharing some of these results and lessons learned, and talking to them about their concerns. These are sort of household name mega site type operations. So I think this work is getting noticed, which hopefully the ESTCP folks should feel good about.
And for our last slide is just an indication of our publications, the framework. As I said, the framework, the final report, and the cost and performance report, we're looking. The Navy has been the long pole in getting these back to the review committee, but they have been approved by ESTCP, but it's Navy getting them through their shop with appropriate language and disclaimers to get them back to get them on the public sites.
And I'll just add that we're really eager to publish some of this work, but we really need fate of carbon data. We need to figure out what happened with the activated carbon and close a bit of a mass balance on that carbon to understand, from what areas it may have migrated and how that influenced our results. So it's really, it's really incomplete before we have the picture of the carbon.
And lastly, this has been a really enjoyable collaborative project. So thanks to the ESTCP group for funding us. It's been a good one. Thank you. That's all we have.
Thank you, and thank you for taking the time to give us the webinar. Are there any questions at this time?
Yeah, this is Steve. I wonder if we can go back to slide 18. I was scratching in my head as well when I saw that.
Yeah.
Well, I guess there were...
This one, right?
No, the pour water. I think it was 18.
I think I'm on it.
The PCBs in pour water.
No, go back one more. It has the dashed lines. There you go.
Yeah.
Yep.
I guess a couple of things struck me looking at this is, why, in most cases, at least in aqueous environments, pour water in sediments, real sediments, has been a very good indicator. It's not here. And I'm also wondering why, and I'll bet Charlie and you, Paul, have some ideas. Why did sediment appear to perform, I won't say poorly, but not as well as aquablock? Was that partly because sediment was applied lower down in the canal and it was more susceptible to movement because of the tides? Or I'm just curious, what are your opinions on those two issues? Why there's such a big difference?
Yeah, I think it's all about the carbon and whether the carbon is there or not. And so there were some differences in these. The sediment was applied by Charlie and Upal in the field, and then and then the control sediment for this study was sent to us from them, as opposed to the 6 and 10 month applications, which were collected by our team. And I think that there's two explanations. One is that there is carbon migration, and that has to do with the physical location on the site. It also has to do with the flows that were experienced and the time that it had to mix into the soil and be stabilized. And we don't have that carbon fate nailed down yet. And then the other thing that was a big factor is the significant change in PCB concentration with depth. And I think that the subsequent samplings at the sedimite plot were conducted with a grab approach that was deeper than the sedimite that was collected as the control that they collected, which was a superficial sample. So what we've done, what we've been working on in the lab since then, in our spare time without any money, is getting profiles of carbon and PCBs, total PCBs and pore water PCBs over depth. And that's the kind of information that we need to understand this. But I think it's certainly not the case that, it all has to do with the carbon delivery and the ability of that carbon to stay put.
I think there's one other thing to add to that too, Steve, is that the sedimite and the aqua block we found out later in the game were applied assuming a slightly different bioactive zone. So we applied assuming 3% activated carbon in the top 6 inches. I think they were working with the 2 to 3 inch mass balance, yet the samples we're presenting here are all 6 inches across the board.
Okay.
So in essence, we may have more carbon than they may have, because we were dosing the top half foot, they were dosing the top couple inches. Yet these samples took that 4 to 6 inch horizon and mixed it in.
And how deep into the sediment was your palm placed?
The palm was put in the sediment after the sediment was collected from the field, brought back to the lab, and placed in a slurry with palm.
Yeah, Steve, this is
slurry. Okay.
This is where I get the question of, so you're taking a 6 inch core back, but let's say your top 4 inches or your top 2 inches is only what's treated with the activated carbon, right?
Yep.
So then you're looking. Your difference in pore water concentrations in the interval that's been treated is going to be much different than the pore water concentrations in the interval that hasn't been treated. Did you guys mix that whole interval then in the lab?
Yeah, in the case of the data we're looking at right here, those were mixed samples, and so essentially that artificially mixed the carbon into that 6 inch depth as you're referring to.
Okay.
So we did not do that with the core samples that we collected. We kept those discrete, and we sliced them into discrete sections to analyze.
We're working on a similar kind of large-scale pilot study with the Navy in Pearl Harbor right now, and that's been recognized. So the bioaccumulation studies are being done on intact cores, in essence, rather than homogenizing the cores and then running the accumulation studies.
And what we have proposed for follow-on work is to collect cores, to conduct bioaccumulation tests on intact cores, and then to section those cores after the bioaccumulation studies to understand how much black carbon is present at different depths, and what the PCB pore water profile is at different depths, to eliminate the heterogeneity aspect and the changes with the depth.
Okay, thanks.
John, this is mostly freshwater at this location, right?
Freshwater tidal, correct.
So you didn't do any field tissue baseline and post because you wouldn't see much difference in the 10 months, right?
We did not. There are, from the CERCLA study, some finfish data available. It would be interesting to look at those, although, again, we were kind of working outside of the channel.
And I think there wasn't enough sample, and there wasn't enough tissue present in macroinvertebrates in order to do that at the site. Isn't that right, John?
That's correct.
No amphibians there, John?
I think there are. I think, especially in the upper wetland, there are frogs, and collecting adult frogs or tadpoles might make sense. It's kind of hard given the nature of the way we did our program here.
Well, I don't think you could control it based on the size of the plots, but I'm more thinking readily on a large scale.
Yeah.
But on a large scale, yeah, no, I think you're right. That might make sense. And they can be pretty good indicators.
Is the bigger issue here more the size of a pilot scale, loss within, these small plots that were done versus being able to see how it works in more of a larger scale active system?
Yeah, I think that's certainly an issue. And initially, we had planned for large, single large plots, and I think it was, I don't know, you probably have a better memory than I do, John, but I mean, it was somewhere in the discussions with the sediment review panel that we switched to multiple replicates of smaller plots.
So that's your way of throwing it at us?
No.
What were you trying to say, Ken?
I said I don't quite remember. It's a little fuzzy, but
okay.
It did change.
I recall that was definitely Tim's recommendation.
Since he's not on the call, we can say that.
Well, it did give us some statistical power, but it introduced some other levels of uncertainty, I guess.
Right.
Yeah. Yep. Exactly.
Are there any other questions? No? Okay. Well, again, I wanted to thank the project team for providing the webinar today and taking the time to prepare and present it. It is very helpful to us, and we do really appreciate it.